This week on the Galactosemia Podcast, I talked to Dr. Synneva Hagen Lillevik. I've been wanting to have this conversation for a long time, and that is because when I first started this podcast, I came across a research paper called Harnessing the Power of Purple Sweet Potato Color and Myo-inositol to Treat Classic Galactosemia. Essentially, it was a study that was published in 2022 where they tested the effectiveness of purple sweet potato color to treat mice that had galactosemia. And they found very positive results in the ovary, the liver, and brain, as well as improved fertility in treated mice that had the knockout GALT gene. This study was done by Synneva and her colleagues, and it's something that I don't think gets enough attention. So I was eager to talk with her about it. We've actually been giving Elliot purple sweet potato ever since I read this study, and honestly, after having this conversation, we're going to continue doing so. Another big area of study for Synneva has been primary ovarian insufficiency, POI, which we know affects many, if not most, females that have classic galactosemia. It's a topic that, quite frankly, we have not talked enough about on this show. So the first half of the episode is dedicated to POI. The second half is when we talk about purple sweet potatoes. It's a really interesting episode. Before we dive in, I hope everyone had a wonderful week of GLOW, the fun run fundraiser for the Galactosemia Foundation. I talked to race director Jamie Hegel, who said that the donations amount increased from the previous year and that there were about 350 registrations. We had a great time in Los Angeles. It actually poured rain for the 3 hours that we had reserved the outdoor space for the event. I found that pretty fitting because if you're familiar with LA weather, pouring rain is a pretty rare occurrence. So how apropos that we would have this rare rainstorm during the very hours in which we champion those that have this very rare disease. At any rate, we did run, we walked, and I hope you were able to do the same wherever you are in the world. We had a couple new signups to the galactosemiapodcast.com, so shout out and big thank you to Danielle Kelly, Christy Gelfa, and Charlotte Swingle for becoming our latest supporters. They get these episodes at least a day early and completely ad-free. If you'd like to sign up and support the show, you can actually get a month free using promo code GLOW at the galactosemiapodcast.com. Okay, let's hop into it. One of my favorite conversations that I have had on the show thus far. Please enjoy this episode with Dr. Synneva Hagen Lillevik. Today, I am very excited to welcome Synneva Hagen Lillevik to the podcast. Dr. Hagen Lillevik is a registered dietitian who recently completed her PhD at the University of Utah, where she studied the pathophysiology of primary ovarian insufficiency and classic galactosemia. She now works as a nutrition T32 postdoctoral research fellow in the Department of Pediatrics at the University of Colorado Anschutz medical campus. Dr. Hagen Lillevik brings a very unique perspective because she has both a clinical experience in treating galactosemia patients as a dietitian and research experience developing potential new therapeutic approaches. In particular, her PhD project involved studying purple sweet potato color as a potential galactosemia intervention and has also done research in primary ovarian insufficiency and galactosemia. Welcome to the show, Dr. Hagen Lillevik.
Thank you so much. I'm excited to be here.
I have to say, the first time I came across the purple sweet potato paper, I read as much of it as I could understand and started immediately giving our son purple sweet potato. So your research paper has somewhat changed the way that I feed my son, and my wife feeds our son, because we try and include purple sweet potato so much. And I'm so excited to talk to you about that today, as well as POI. But from the start, I always ask the same question. I like to start off with this one because galactosemia is such a oftentimes confusing rare disease. When you meet somebody who has never heard of it before and you want to tell them what it is, whether it's like, hey, this is what I study or what I do or what I'm interested in or the research that I've done, how do you describe galactosemia to someone who's never heard of it before?
Yeah, absolutely. People generally glaze over when you tell them, what what types of patients you're seeing in clinic or what you're researching. So I say that I studied a very rare disorder of metabolism it's an inherited disease, essentially. So individuals with this disease have a genetic change that makes it so they can't process a carbohydrate, which is called galactose, in their body. And the only treatment is really dietary restriction of galactose. And so as a dietitian, we help people have a well-balanced and healthy diet to get them what they need while keeping galactose out of the diet.
That's a great explanation of it. And I find it interesting. I love asking that question just because whatever kind of looking glass you're looking at it through, whether it's a patient or a dietitian or Dr. Barry, who's the smartest doctor I've ever met in my entire life. And it's like the answers, vary so widely, but they all are kind of getting at the same thing of like trying to describe this kind of hard to understand rare disorder or disease. You and I were talking before about the conversation that I, as of this recording, just had, but as of the release of this episode will have come out last week for the people listening. But I had a conversation with Sandy Van Kalkar, who is kind of the go-to dietitian when galactosemia, and a lot of the great work that she's done just in terms of galactose levels and studies. And I just think it's so interesting that you have this dietitian background, but you also went into research of galactosemia. What was that like?
What's the connection there?
Yeah.
Well, so when I finished my master's degree in nutrition, I was looking for clinical positions, and so working with patients in and out of the hospital, and I got a position in they call it the Metabolic clinic, but it's essentially the group that sees all of these different rare disorders of metabolism. So, in addition to galactosemia, there's things like phenylketonuria, which is a disorder of protein metabolism, PKU. And that's, so these types of diseases or conditions, I guess, we would see in clinic. And the primary treatment for most of them is diet. So that's why they have a team of dietitians and to see patients. And as I was working in this clinic, I saw a lot of different conditions where diet was the primary treatment, and lots of different things. But with galactosemia, it was interesting because, I don't know, relatively speaking, the diet, while it feels really restrictive, it's not as precise, I guess, as some of the other disorders, like with the protein metabolism, where, there's sort of this chronic over time problems with galactosemia. But, some of the other disorders, if you go off on the wrong, with the wrong foods, you can have some really severe, end up in the hospital kind of thing. So, but I was just not seeing as great outcomes for patients, even knowing they were perfectly, doing great with what the dietary recommendations are. So I always wanted to go back and do more research, and there were a lot of unanswered questions about galactosemia and, what was happening on a cellular level. Like I said, even with individuals who were following the dietary recommendations and treatments perfectly. So there, when I was looking to go back to school, I found a lab that was connected with our clinic at the University of Utah that Dr. Kent Lai was studying galactosemia, and it was a really good fit. And I think for the ovary, looking at the ovary and the ovarian complications that happen for females, I've always been interested in women's health and health outcomes, and I had also had some pretty some patient interactions that were pretty formative for piquing my interest in that area. So
Interesting. Without disclosing, obviously, any HIPAA or anything like that, can you tell us anything about like what those experiences were like?
Yeah, absolutely. I just remember being in a visit with a young woman. She was maybe 14, 15 at the time, and her mother. And they were just feeling really desperate about, she hadn't really started her period. Her menstrual cycle was delayed, and she was on birth control for hormone replacement. But there was just this sense of anxiety and urgency and sort of almost desperation, I guess, of what the future was going to look like. Because even at this young age, this patient had already been, they had been tracking blood levels of follicle stimulating hormone and anti-Mullerian hormone, AMH, when this patient was younger, and up to then. And, biochemically, things were looking like she was having some premature ovarian insufficiency, and then having these clinical features of not having a consistent period, and just kind of what that looked like for future if this individual wanted to have a child. And we didn't have the answers to that. And so that experience, it was a hard visit, and just the parent, the mother's perception and sort of her questions, and then her child, what that meant to her as a young female, I think was really striking to me.
And that led to whenever you found Dr. Lai and the University of Utah that were studying galactosemia, and you joined up with them, the decision to focus some of the studies into POI and its complications with galactosemia.
Yeah, I mean, there were a couple of things with that. Definitely that experience in clinic was part of it. Dr. Lai was a really great mentor in that he kind of laid out some, gaps that his lab was trying to fill, and, said, what are you interested in? Because ultimately you're going to be spending a lot of time on this. So, really make sure it's something, that you're interested in. And then also, I think studying the ovary too, it can be a good place to start, in that they had a mouse model that the mice with the gene with the gene knockout, the knockout mouse model, essentially, they showed that they had fewer pups and really like a sub-fertility characteristics, where they would, have just a couple litters and they'd have less pups than the animals that didn't have the genetic change. And so he had a really great model to be able to study this. And there's just a lot of unanswered questions about primary ovarian insufficiency and galactosemia. Does it start in utero? Is it progressive after birth? So I kind of set out to answer some of those questions on a molecular level and on a follicle level.
And that's like, when I talk to Dr. Judy or any of the researchers who are doing work that matters like this, it's comes from such a place of caring and like empathy, and I think that's really amazing. And you studied at University of Utah. Now you don't live in Utah. What are you up to now? I know there's a T32 program, which I asked ChatGPT about. That said that's an NIH funded thing. Tell me about what you're up to these days.
So I when I finished my doctorate with Dr. Lai, I went on to do what's called a postdoctoral fellowship. And I shifted gears a little bit in subject area, and I'm actually studying gestational diabetes, so carbohydrate intolerance in pregnancy. that first shows up in pregnancy. And there's various reasons why I'm looking at that, but I'm still in, women's health and diet and carbohydrate metabolism, which galactosemia, galactose is a sugar, which is a carbohydrate, and there's quite a bit of overlap between these metabolic pathways. So.
Yeah, you were able to harness that metabolic pathway knowledge and experience that you had and move it into the similar space, it sounds like.
Absolutely, yeah.
I want to make sure we hit two large topics with you. And I went back and forth on which one to put first because one of them is primary ovarian insufficiency and the other one is purple sweet potatoes. So we're going to start off with POI, primary ovarian insufficiency and galactosemia. And I know that was a major focus of your PhD work, as we just talked about. We had a listener reach out actually asking for an episode on this topic specifically, and then we've got a question from her that we're going to answer or try to answer in a bit. But can you just explain what primary ovarian insufficiency is and how it affects women in galactosemia on kind of just a 30,000-foot view, and maybe then dive a little bit deeper into these areas?
Yeah, so in general, I think the official definition of primary ovarian insufficiency is, the absence of normal menstrual cycle, essentially menopause before the age of 40. And it's characterized by different things. It's a little bit different in galactosemia as well, because usually you're seeing clinically significant evidence of POI quite a bit earlier with females with galactosemia. Research has shown that it affects up to 90% of females with galactosemia. So it's a pretty prevalent complication. And there's different ideas of why that is, mostly kind of the microenvironment of the ovary. Like the metabolic environment, I guess, of the ovary is very sensitive. So in general, females are born with all of the eggs that we will have throughout our life. And those eggs, they start out in what's called a primordial follicle. And they're developed in utero. So when the female is inside their mother's uterus, around 20 to 24 weeks gestation, those primordial follicles are formed and actually start kind of their life cycle. So kind of the life cycle of the follicles in the ovary, they go through different developmental stages. And this is in normal ovarian function as well, where, every month you're losing like a bunch of follicles. And so essentially then by the time you're 50, which is kind of the average age of menopause, those primordial follicles have gone through their lifespan and are essentially gone. And so that's kind of what primary ovarian insufficiency is looking at, like either an accelerated loss of those follicles or actually something that makes those follicles not develop normally. So there's kind of two different theories of, those follicles never matured, they die off rapidly, or there's kind of a burnout hypothesis where they activate to go through different stages prematurely and too quickly.
They drop out quicker than they normally would. Are both of those hypotheses like at play and like they could both be possible, or do we know that they both happened? Is there any insight into like which one is probably more likely?
So that was one of the things I was trying to answer with our mouse model. We had a mouse model of galactosemia, which I talked about, where the mice have issues with their fertility. And so I was actually doing follicle counts at different ages. So as early as 5 days after birth, and then through like what we would consider a sexually mature mouse, to look at the differences in follicle types and number. And so really looking at those primordial follicles. And in studies with humans too, that's kind of what they look at, is that primordial follicle count. Because essentially if you have a good pool, you want to preserve that primordial pool for as long as possible to keep the health of the ovary going. I should mention too that it's not all just about fertility and being able to have a child necessarily. The ovary has a lot of metabolic functions for humans, I guess, or for female humans, as far as like bone density and cardiovascular disease risk kind of increases after someone goes through menopause. So there's a lot of other health, kind of long-term health benefits of keeping ovaries healthy and, more in that timeline. But going back to the follicles, so that was one thing we were trying to determine, is if in our mouse model, are the ovaries kind of showing more of this like accelerated growth and burnout phenotype, or like if that's what was going on in our mice, or were the follicles just dying, if that makes sense, from the primordial stage,
not developing the way they should, not getting to that stage which they should mature.
Right, exactly. So we looked at that through actually just looking at the ovaries, staining them, and counting the follicles. There's a method you can do to like estimate how many follicles are in a ovary based on like sections of an ovary.
Okay.
So we did that, and then we also did a lot of staining for different molecular markers, which kind of gets me into the oxidative stress zone and some of those other things that I know you want to talk about with the purple sweet potato.
Yeah.
So we found some differences in some of those molecular markers. We also stained for some markers that would indicate growth, like cells that are alive and growing and healthy versus cells that are sort of earmarked to be destroyed. So
interesting.
Which
to my unscientific mind means that maybe they mature and then burn out faster.
Yeah. So that's essentially what we found in the mouse model. So, there are differences between mice and humans and with the ovary. And so, you can't just extrapolate that. But I mean, it did give us some idea of what could be happening because in human studies with galactosemia, they've done some follicle counts by taking a piece of the ovary out and looking at young ages. It seems like there is a similar amount of primordial follicles, like below age 5. But then as you get older, there's less in females with galactosemia. So,
got it.
So
what does that inform then on? Like, we've learned that, we know that maybe they're burning out quicker. Let's say that's our thesis. What's then the thing that we take from that, and how does that inform other stuff?
So trying to find a way to slow down the activation of those follicles. So in Kent's lab, they did some other studies as well that actually used a therapeutic. It's a drug that's not really been tested in humans. It's a drug that acts on some of these stress response pathways. It's known to kind of slow down the stress response. And we're kind of getting into the weeds here a little bit, but basically what I found with my work and then work before is that in the ovary, those follicles are really sensitive to these stress signals. And so any sort of, strain on that can either cause something to start growing and die or to die on its own, if that makes sense. So we were trying to tease out kind of what that potentially could look like. But some of the work that Kent and Begina did prior to my study was using a therapeutic drug compound, essentially, with the mice that is known to sort of tell the cell to slow down and to stop translating protein and going, kind of just to chill out. I guess you
stop
could
panicking.
Say.
Yeah, it's okay. Like,
yeah.
So do I have this right? And we'll talk to Dr. Lai and Bajina as well. I want to say that on the podcast. We're going to have them on a later episode who you worked with. But essentially what you're saying is these follicles, they, like you said, they're very sensitive to the metabolic pathway and the metabolism and things that are happening in your metabolism. And we know that galactosemia has to do with issues in the metabolism in terms of there are variants that make the GALT enzyme not work properly, which puts stress on your cells and your tissues. And so these follicles might be receiving that stress and therefore speeding up this process of developing and killing off the follicles in a way that they should not. And so a method of slowing that down might be to just like tell them like, hey, chill out. Like, this isn't a panic situation. Like, there's not so much stress that needs to like be responded to.
I think that's a great summary.
Great dumbed down way of thinking about it. Okay,
yeah, and I'm just rolling around all these like molecular things in my head that we've talked about and all these conversations, but that's like a perfect way of describing it. I kind of think of it like, okay, you have this metabolic block, which is like a log jam in the river, because the enzyme's not working. So you have all this stuff coming in, which in the case of galactosemia is galactose, and it can't get past. And so you have these buildups of Gal-1T, for example, galactitol. Right. And those have implications in the cell. You know, they may inhibit other enzymes that need to do their job, which we haven't even talked about myoinositol, but that's one of the theories with that, is that Gal-1P inhibits an important enzyme for regenerating that in the cell. So the theory would be if you supplement with that, you can replete some of that. But then, and then, you know, with galactitol, that is really kind of cell toxic to the cell, and it creates a lot of stress and free radicals. The other part of this is you've got this jam here, but then what's happening on the other side is you have a decrease in whatever it is you're trying to make, which in this case is UDP, galactose, which is really important for a bunch of other cellular reactions and glycoproteins and things like that. And so you kind of have all this junk that's building up and then less of what you need. And so that causes stress in the cell.
Yeah. And the downstream effects of that can be things like primary ovarian insufficiency, or we've talked on this show about like seizures, and 25% of people with galactosemia have seizures. And it's like these things, these domino effects that we are kind of like, does that have anything to do with galactosemia? Doesn't have anything to do with galactosemia. Some are more obvious, some are not as obvious, but what you're talking about is this downstream, like, well, yeah, it's really hard to tell because we got this log jam here, and this is something we're trying to look at over here, but we don't really know what the correlation is and the connection. And it's starting to make more sense it's just so hard to like test for all these things.
Right. And I think that's one of the reasons, well, with the ovary, it's always the question, you know, is the GALT enzyme, does it have a function in the ovary specifically? And I couldn't quite figure out how to answer that question with what I knew of ovarian biology and just coming into galactosemia, you know, from a molecular level. But we do know that in other forms of, or other reasons why people have POI, that oxidative stress and that sort of micro environment is really important. And so that was one way to look at it. And then also with their studies where they gave the mice this stress reducer, essentially.
Did they see that helped?
Yeah. So
let's approve that medicine that's never been proved for
I know,
humans.
and that was a whole topic too. And I mean, I think with drug development and discovery, it's a whole process, and it's really tricky, and you have to do so many, you know, it's years and years and years of work just to show.
So much money, and it's like, and I would imagine from the research level, and I'm just finding out, like, I'm kind of dipping my toes into like pharma, because I'm talking to these gene therapy companies, and from from a research level, like your job is not to develop the drug. Your job is to do the research to get us to a point where it's like, is this viable or not? To do the researches and the tests.
And is it safe?
And is it safe, right? And that's so important. And then you have this pharma industry that, first of all, there's like so many drugs and diseases out there, right? So how are they picking what they're picking on, and what's more beneficial to their bottom dollar and going to make more money and all of this stuff? And then it's like, to be able to say, well, there's this one drug that maybe has an effect on primary ovarian insufficiency in women with galactosemia. So probably 1,500 people in the US, like, to develop that drug and what it takes for a drug trial, it might not just be viable. And so then, my brain kind of goes to, well, then what are you to do? Because you got, like, the 1,500 women are still being affected by this in the US. So, like, it's such a hard position.
Absolutely. And I think that's why nutraceuticals, or naturally occurring products, are appealing. In other inborn errors of metabolism or other genetic metabolic diseases, there are, nutraceuticals used in different things, or single amino acids, or, some of these different types of things. I mean, we can launch into the purple sweet potato thing.
I want to get there. I want to talk first. What are the current approaches to managing POI in women with galactosemia?
Yeah. So it's kind of symptom management at this point.
Really?
There is nothing to prevent it, essentially. I mean, even individuals, like I said, who are following their diet and doing everything right. If a female or a girl is getting to be of age where you would expect some of these, to have a menstrual period, or some of these secondary sex characteristics like growing breasts or things like that, and that's not happening, there's hormone replacement therapy. I think clinically, what, at least a couple of years ago, and I don't think anything's changed, I mean, I could be wrong on that, but I think clinically the approach is to refer to an endocrinologist, a pediatric endocrinologist, to sort of manage these hormones. And yeah.
And one of our listeners, Charlotte, wrote in and had a very thoughtful question about fertility preservation for girls with classic galactosemia. From what she has read, it seems that the egg cryopreservation is being recommended even for prepubescent girls, possibly involving stem cell-like eggs. She was a bit unclear on what the specifics were, but she wanted to know, like, is it true that some CG girls have eggs cryopreserved, but since they're still very young, the IVF using those eggs hasn't yet been attempted? Yeah,
so it's a really good question, and I think it's a really interesting question. So from what I know, so far this has only been done in research studies with a very small number of girls with CG. I don't think it's being recommended universally. Across clinics. I like I said, I can't really speak to many clinics. I know in the clinic in Utah, they're not recommending that necessarily. So, and that is true that this was done quite recently, within like the last 3 years. And so the participants, the young girls, have not reached the age where they might be putting that tissue, that cortical tissue, back in to have a pregnancy. I think, going back to what we talked about with the health of those primordial follicles, I think there's a lot of unknowns as to whether that tissue, even if it looks fairly normal in young girls, whether that tissue is normal on a molecular level, or if sort of those metabolic hits have happened in utero, before the child was even born. I just think there's not enough information about that.
And Charlotte saw some references to an age as being as young as 6 months, which seems surprising. What kind of things do you think should be considered when now we're thinking about egg retrieval in very young girls, or what age they are when that kind of thing can happen? It seems like there would be a lot to consider.
Yeah, I think from an ethical standpoint, I mean, those are pretty big. I mean, it's a surgery. If it's your own child, and there's always questions too, whether that child is going to be interested in having a child later. I think there's a lot of questions about that. And then also just the health of the ovary. And I don't know enough about those fertility preservation techniques. I know it's more common in individuals that are maybe going through cancer treatment at very young ages. But their follicle health might be a little bit different than somebody with galactosemia. And I think we just don't really know what that's going to look like. Right. So,
again, another kind of between a rock and a hard place of, when you start thinking about egg retrieval in girls that young, even if they're doing them with other girls with other conditions like childhood cancer, how much are their follicles different? And how much of an effect does that have on somebody with galactosemia? Does it even work? Is it safe? But at the end of if it does work, you're not going to know without trying it. So it's so hard of a place to consider.
Yeah, I think it's a really tricky question, and it has a lot of different facets to be considered. And I guess a clarifying question, when you're talking about egg retrieval at 6 months, are you talking about taking actually a piece of the ovary, like in cryopreservation? Because they do more like a tissue where they take a piece of the ovary that then they can transplant essentially back on to the ovary when that individual is wanting a child.
I don't know what I'm talking about, to be honest, and clear. I would say, is there one way that's better than another, would I guess be the next question?
Well, I think in galactosemia, they've done that tissue, the cryopreservation. I don't know if they have, given the young girl hormones to actually, have an egg release. I think there would be some major there's ethical issues either way, but right. I don't know about that, but
got it.
I wonder, that's maybe a follow-up question. Yeah, I wish I could speak more to that, but just from knowing what I know about the molecular changes in the follicles, there's a lot of unknowns about what that tissue would actually look like once you put it back on. So.
Yeah. And I think, even having this conversation is important. And this is you have to have a lot of conversations to get to a point where you're starting to become informed and you don't have all of the answers for fertility. And Charlotte it's obviously something that she's very thoughtfully considering of what should we know about this topic. And I think these are the conversations that need to be had. And in a rare disease community like this, it is so hard sometimes to find the information that you want to find, whether it's because it's just not out there and does not exist, or the people who know it are too busy doing research on other things. And, I think that's the reason I started this podcast, was just so I could get people like you on the line and talk about these things. So we might not have all of the answers, but I think, it's worth continuing to look into and seeing if maybe there is more information out there on fertility, or what type of retrieval they're doing. And, I think it's worthwhile to keep going.
I wonder about talking with the authors of the research studies that they've done in galactosemia. They likely have a different, or, they might have a perspective that's valuable on what the work is that they're doing and what they're seeing,
right.
From a molecular level. Again, I haven't done, this has been in mice, but the work that I've done, so.
Yeah. Well, and you mentioned reaching out to the authors of the research papers, which is exactly what I did when you received my cold email of, hey, can you talk to me about this purple sweet potato color research, which is what I want to get into now. Because that's what initially caught my attention about your research, was purple sweet potato. And I remember reading it and being like, why is nobody everyone's talking about Govorestat, but it feels like purple sweet potato is a naturally occurring, could have an effect. And I've literally heard no one else in the community talk about it. And that I wanted to do this episode specifically to talk about it. You co-authored the paper called Harnessing the Power of Purple Sweet Potato Color and Myo-inositol to Treat Classic Galactosemia. I will put a link to that paper in the episode description, but can you tell us about what you were looking into? What is this? What did you find? Like, how did it get started? Give me the goods.
I should have journaled on like the whole process, of course, but, yeah. So, and we've touched on this a little bit, but the whole oxidative stress and, inflammatory.
What is oxidative stress? Can you break that down for me?
Yeah, well, when I was talking about the kind of the log jam, and you have this metabolic junk essentially, that has all these other like wide-reaching implications in the cell. And then also with Kent and Begina's previous work, where they really mapped out something that's called the ER stress, or endoplasmic reticulum stress pathways, which is it's a pathway essentially in the cell to either keep the cell alive or to kill the cell. And there are a lot of different things that will stimulate the cell to sort of activate this stress response. And one of the central markers is this protein, EIF2 alpha, which I was talking about sort of previously with this drug that actually acts on this. But what it does is it kind of tells the cell, okay, let's speed up our growth and our protein synthesis, or let's shut it down and be quiet and just hang out, right? And when that protein is active, different things happen, and there's different downstream effects. Cell death is essentially the one that everyone's mostly, but we have these processes going on in our cells all the time. You have kind of a balance between, inflammation, oxidative stress, and then these anti-inflammatory, stress-reducing pathways that are in balance. But one of the theories of how this is working in galactosemia is that you've got an imbalance where you have a lot more of this stress, these reactive oxygen species that cause damage to cells and different pathways and things like that, based on kind of that metabolic junk. And also the lack of UDP galactose, which
both sides of that logjam, the build up and also the lack of resources.
And that's that's getting into a whole different cell world. But galactosemia is considered a secondary disorder of glycosylation, which is a cellular process. And
I will Google that after we're done talking.
And when you talk to Ken too, he's working on other congenital disorders of glycosylation, and that's more his wheelhouse. But one of those things that kind of activates the stress response is problems with glycosylation. So that was kind of like the way back theory. And then, can we manipulate this pathway, this stress pathway, to cause better outcomes? So, the things that you're seeing, like the seizures or the ataxia, the tremor, primary ovarian insufficiency, things like that. So, because that's essentially the result of all of this cellular
stress.
Yeah, and dysfunction, I guess you could say. Okay, so going back to purple sweet potato. So actually in Dr. Judy's lab, she did some work with oxidative stress in her fruit fly model, and showed that that there was oxidative stress and that you could mitigate some of the essentially death by giving these free radical scavenger. This is probably a very crude description of this work that she did, but she had one of the papers on that in the fruit fly model, and then from there was a
wait, what did she give the fruit fly to lessen their death rate?
Basically antioxidants.
Antioxidants.
To help with some of the adverse outcomes that she
found a chemical that was an antioxidant and
I'd have to look up in the paper exactly what
I'll just text her and ask her.
Just ask her, because like I said, that was probably a very crude interpretation of her work, but that was kind of the one of the papers that then there was a researcher, Dr. David Timpson from the UK, who was really interested in galactosemia, and he did a lot of work on galactosemia. He's since passed, but he had the idea about purple sweet potatoes. And the reason...
What was his idea?
So his idea was that there are naturally occurring chemicals in purple sweet potatoes that are very, very powerful antioxidants and anti-inflammatory properties. They're essentially like, I kind of think of it like a drug almost in the plant tissue that the plant naturally produces, and we see it as that purple pigment, but they're called anthocyanidins or anthocyanins when they're in a slightly different chemical structure. These phytonutrients or plant compounds essentially are known to act on various pathways in the body, and they're an antioxidant. So you've probably heard of like blueberries. Everyone's all excited about blueberries and like the skin and the blue, and those are
antioxidants in blueberries. 100%.
Exactly.
Always hear that. Yeah.
Cherries are another really rich source. There's been quite a bit of work done on strawberries. And actually, I just listened to a really interesting talk about strawberries and anthocyanins. They have slightly different class or different compounds in them that are unique to strawberries. So strawberries are great too, But the reason for the purple potato is the tuber, or like the root, actually makes a lot of this purple, the anthocyanin.
Okay.
And it's a relatively cheap source of a really dense, the anthocyanin compound, that purple color. So it was his idea originally, and I actually emailed with him when I was developing this project to talk with him a little bit about, his work and how we could do it. And he was
he excited to see it kind of come to life?
I think so. I mean, I hope so. Yeah.
He
had an idea to grow purple sweet potatoes in the lab, and then you can, through different extraction techniques, pull out, kind of the anthocyanin, the concentrated anthocyanin content, which is essentially what we did with the PSPC, the purple sweet potato color.
Yeah, so that's interesting to me because I'm just feeding Elliot purple sweet potatoes every day. I'm like, hey, eat these purple mashed sweet potatoes that I found at Sprouts.
Yep.
You used a purple sweet potato color, which was an extraction from a purple sweet potato. Do you have any idea of like what that concentrate kind of translates to in terms of like an actual sweet potato?
Yeah, so that is a really interesting and good question with that. I don't know like numbers wise. So the anthocyanins in purple sweet potato have been used as like a food coloring ingredient in a lot of different like consumables, I guess. So that's another reason why it was attractive, is because it's been used like in juices and drinks and makeup and things, like a natural food coloring. Kent is really great with really wanting to get to the mechanism on things. So, we thought about just doing the purple sweet potato, but there's a lot of other really good things in purple sweet potatoes too, different starches and fibers and other of these like plant compounds that are great for the body. So I think it's great that your son is eating purple sweet potatoes.
Yeah, you mentioned that your family eats purple sweet potatoes all the time.
We do, yeah. We make them into like fries and bake them, and yeah, they're great. It's really changed our dietary intake too, this research. But we really wanted to get to sort of the bioactive compound and really test, like, if we do kind of a concentrated version of this. So we had a company take the same sweet potatoes that you get from Sprouts. They essentially pulled out the purple color, that compound of the potato. And it was interesting because they are more from like a pigment standpoint. Like they extract things and then they make different shades of colors. Because with anthocyanins, if you add acid or base, you can change the color slightly. So they developed a color, vegetable juice color for us. But it was essentially like a powder that was just a concentrated form of this chemical.
I've actually found online, I Googled it, there's purple sweet potato powder.
Mm-hmm.
Is that kind of similar?
It is. I think that's mostly dried sweet potato.
Sweet potato?
Yeah.
Got it.
So like there's a, there's a company that will like dry it and you can put it in your smoothies or, it's like a superfood supplement type thing. I'm pretty sure it's just the sweet potato. So this, of course, I don't know exactly how they did it, but you can there's different extraction methods based on the chemical makeup of the anthocyanin. So you can use different, like, I think alcohol, like ethanol can be one way to like really pull that compound out. I don't know how they did it exactly.
But they found a way to pull out the purple sweet potato color, which is what you guys call the extraction.
Mm-hmm.
And so then you all give it to the mice in the mouse model. What outcomes or improvements did you observe using the PSPC?
Yeah, so one kind of middle step, we actually sent it to a company that makes mouse food, and they added it to the feed, like so the little mouse pellets. So we had like purple pellets that the mice got, and then we had the control diet, which they didn't have the purple in there in the pellet. So yeah, we did that, and then we did a fertility test basically. So we had mice that were eating the purple sweet potato, female mice. Once they were done nursing, once we weaned them from their mothers, they were on the purple diet until they were old enough that we could pair them with a male. And then we measured the mouse menstrual cycle, which sounds really funny, but to see if, it was like, because that's kind of a marker of like if the ovary is working.
Right.
And then, so we did that, and then we paired them with a male, and then we timed, how long did it take them to get pregnant? Because mice get pregnant pretty quickly. And so we timed, how long it took them to get pregnant, and then, how long to have that first litter, and then how many pups were in the litter. And then we compared it to the mice that were on the control diet. And these were mice, our knockout mice that had the deficiency in
Galt enzyme.
Yeah, exactly. So then we measured their fertility basically, and we measured a lot of other things too. We looked at brain tissue, we looked at liver, we looked for signs of toxicity. So like if the mice looked like they weren't doing well or having any problems with the amount of the purple sweet potato that we were giving them.
Right. Are you giving them too much purple sweet potato that they're like becoming a purple sweet potato or something, Well,
and another cool thing about using anthocyanins is I have never been able to find anything that says that there's an upper limit to the amount that you can give. And the amount that we gave the mice was based off of some studies that were done with purple sweet potato extract. And they weren't knockout mice, but they were mice that were given galactose. So this idea has been kind of floating around in different ways in different fields almost. So
yeah.
But yeah, we saw the pups or the mice that were treated, they had more pups per litter, and then I followed them for like 5 different litters, and the pups or the mice that were not treated stopped having litters after like the second or third round, but the ones that were treated continued, 5 litters. So it was pretty exciting.
That's super exciting.
Yeah. And then we looked at the ovaries themselves, and they had more primordial follicles. They were different mice, but they were mice that were treated. They had more primordial follicles, at that sort of mature age, whereas the ones that were treated had less follicles in their ovaries at the same age. So
what year was this, by the way?
2021, 22.
Okay. So it's 3 or 4 years ago.
Mm-hmm.
And then you graduate, you get your PhD. This paper comes out. To me, this is like so exciting. And it's like, holy shit, like purple sweet potato could be like a huge benefit. But like what happens?
What happened? Yeah. What happened? There are so many different issues with how to like get this to humans, and
What are some of those issues?
Well, safety is the one thing. And so actually, Begina just published a paper doing a safety studies on purple sweet potato, where she gave, different amounts, and then they tested different things. And it seems like it's safe. So with this extract for the mice. But, it's hard when you have a child, you have that question, is it safe that I'm giving my son purple potato every day? I think it really goes back to, I think it would be great if people were doing this, if we could sort of look at, do like a natural history study where people are supplementing and what they're supplementing and how often and, what are the outcomes, as far as looking at the ovary and purple sweet potato. I mean, it's something that we've talked about, like, well, could we give it to young girls with galactosemia, follow them till they're 14 and see if more of them have normal periods, you can't really take the ovary out and look at it. So we've talked about some of this stuff, but we haven't really found a way to really look at it. There have been some clinical trials actually with purple sweet potato juice, where they juiced the potato and gave it to adults. And they were looking more at metabolic markers, liver function tests and blood glucose and things like that.
Do you know what they found?
I think it was an improvement.
Yeah. Well, and my question, I guess, comes from we buy these purple sweet potatoes. I buy them at Sprouts. They're this big. They're huge. They're huge potatoes. So we take the skin off, chop them up, we steam them, mash it. My son's 8 months old. He's getting 6 bites before he's it's all over his face. It's everywhere. I don't know that that's enough to do anything to his system, as much as if there's an extract or a concentrated version that I'm hey, drink this out of your cup, and it takes him 4 sips and he's all of a sudden got all of this antioxidants that are relieving his cells of stress. I would much rather, one, I'd much rather do that than give him the purple sweet potato, but 2, I'd much rather do that over giving him some chemical pill or something that we're testing in a chemical trial that it seems pretty low risk to me to give him purple sweet potato.
Yeah, that was part of why I wanted to develop this project, because when you have nothing nothing that you feel like you can do and have any control over, when you have a child who has something not unusual, but something that you didn't anticipate having to do, I think it gives some power to parents to be able to do something with the diet.
Totally.
I think, the question of safety with the extract, I've wanted to just try it myself, and see if I can get my hands on some of it and then just see what happens,
Well, the question is it's not the most safe thing, but I wonder, you guys went to a lab, you said, can you do this for me? Did they ask for credentials on research and stuff, or could I do that? Could I go to the lab and be hey, give me this extract, and I could take it myself and see, is that doable for a consumer, or is that purely research things that are required by the labs?
I don't know. I had a feeling you were going to ask that question, because I've been whoa, should we just I don't know. We, when you go through the university, there's chart fields and all these different things. But the one thing that I do worry about a little bit is, I mentioned when you do it in a lab and you extract, we get a form that says it's safe, for the animals to eat.
Right.
Because you have to go through pretty rigorous, you can't just be giving animals, whatever, you have to go through a review process for the animals as well to make sure you're not harming the animals. So, we got something that said that this was safe, essentially. But I don't know what they used. So, if they use something like methanol, that's pretty toxic. But I don't know. I was telling Kent earlier today, actually, that when I was figuring all this out, I took a bunch of sweet potato and tried to like let it sit and like get the anthocyanins out and like extract it. And I juiced it with like a juicer. And I mean, ultimately we went this route to get more of a purified compound, because we did actually test it in cell culture as well to see, you know, if we could reduce some of those reactive oxygen species in cell culture.
Did it?
Yeah.
Did it?
Yeah, I did. And so we were looking at different concentrations. It was more like a proof of concept, because then, you know, when you're doing something in a cell dish, it's really different than actually giving it to the animal. But so we didn't want to put like a bunch of like that starch stuff that's in the potato, you know, other parts of the potato in the cell culture, because it's really delicate with the
that makes sense.
Everything. So.
Well, when you did like you let it sit out and you tried to juice it and stuff, did you test the anthocyanin levels on those?
No, I didn't.
Can you test that?
I was just trying to see if we could if I could do it, you know? We didn't. And actually that was a tricky thing too, because there's all these methods for actually testing anthocyanins. And we found someone who was willing to run our extract in their lab, actually in Colorado. They're on our paper. I think they're the third author on the paper, maybe second author, but it was great. So they used a method to kind of help us estimate what anthocyanins we had in the extract to make sure there was something bioactive in there. But, you know, if I were to do this study again, I would do a different technique and really have a rigorous testing of that.
Well, and my question is, could I hit that person up hypothetically and pay them to not do extract thing, but could I pay them to like juice a purple sweet potato and do that same test and let me know how the levels compare between the extract and a juice?
Yeah, probably not this same person. I don't know if they're still in their lab, and I should get in touch. But there are.
But like, is that from a research standpoint, from a standpoint of someone who's much smarter than I am? Does that check out?
I mean, you know, there's politics and everything, but you probably somebody could. I mean,
I'm gonna bypass all the politics. I'm not part of a system. I'm a podcast on my own here doing random things.
Yeah. No. I don't know. I probably should.
Okay.
Yeah, I mean, you could. People test things in their personal lab friends all the time, you know, but.
Right. All right. Enough said. You don't have to go on record.
Yeah.
Let's stay tuned to the podcast. We'll see what happens. Who knows? So purple sweet potato color has an effect. It seems very positive, but it kind of like after the research paper, you moved on to a new project. It sounds like Dr. Lai and Bajina are still doing some things, but there is probably some potential out there for purple sweet potato to make a difference. And even if it's not specifically make a difference, like you said, it's probably good just to eat it anyway.
Yeah, like when you're talking about, you know, I'm a dietitian, I guess, and anytime we can eat more colorful fruits and vegetables and help our kids eat those foods and enjoy those foods, that's great, you know. And it seems from as we were talking about before, you know, with Dr. Van Kalkar's work, that fruits and vegetables are safe.
Yes.
In galactosemia. And so, I mean, we should all be eating more fruits and vegetables, basically. So, so there's no harm there. I think there are some questions when you get into like a really consolidated form. I'm interested in moving that forward. I haven't really been able to figure out professionally how to do that. Just the way that research and more clinical trials. The Galactosemia Foundation has been really generous. They funded my project.
Oh.
And I know they funded Bajina's follow-up on the safety project. So they've been really great. So yeah, I don't know what the next steps are, but
all right.
Pretty
Stay
cool.
tuned. Yeah, I love it. All right. Well, we've been chatting for a long time, and I don't want to take up too much of your time. I want you to let you have your night as well. But just in both as both a researcher and a registered dietitian, I think you've got a very special view on nutritional management of galactosemia. And I'm just wondering, I guess from a nutritionist standpoint or dietitian standpoint, and also a research, how have you seen the diet evolve like during your time in this field? And do you see it from a specific point of view as a dietitian going anywhere? Like, are you staying in touch with what's happening in the field, or are you kind of off on your own doing other stuff these days?
There's a couple questions in there. I guess you talked about the evolution of the diet. I think right when I started as a dietitian, it was newer information that, all fruits and vegetables were safe and there's benefits of including those in the diet. And I don't think since then, I'm not aware. I kind of keep, one ear or one eye on what's happening in rare disease because I just really, I love it. I mean, and I love the patients that I've had and the families, and it's just been really a great experience for me as a clinician. And then obviously for my PhD work, working on galactosemia, that's been really great. But I don't think there is much, there's not much new out there, I guess. Yeah. I mean, other than maybe this whole purple sweet potato thing,
it's pretty, I think we're breaking some ground.
And some of the supplements, I think it's pretty cool. I mean, but I think, and I said this in the beginning, I think it applies to lots of different diseases. I mean, so I'm researching diabetes right now, and there's a lot of similar stress pathways and mechanisms and things like that. So I think it does still, it's very relevant. And then the second part of your question, you asked about the evolution as a dietitian.
Well, I was just asking about, how much you keep an eye on it, but you said you keep one ear kind of tuned in. One question I had was just as a dietitian and a researcher, if there's a question about galactosemia that you would most like to see answered in the next decade, what would that be?
I don't know. I think there's some exciting things, which you've talked about on the podcast, coming down the line. I think too, so part of my work right now is I'm looking more into some of the psychosocial aspects of disease. And I would like to give families some stress relief and some hope with all this. And so I don't know if that's through diet. Since diet seems to be, kind of one of the treatment things, and obviously that I'm interested in. I mean, I think we can manipulate a lot with diet and metabolism with, as I was, talking about the purple sweet potato and whatnot. But I think maybe combining, some of the gene therapies and the other therapies with diet to kind of give parents different tools. I think I would like to see that, because really going back to that first story I shared, just the psychological distress that parents, and I am a parent myself, it's it affects so many parts of our lives. And so I wish that we could address some of those issues, and maybe through some of these treatments and these therapies, because,
Well, if I could just say, you mentioned stress relief and hope for parents and patients, and I think that a lot of these things being developed with that intention in mind is so important, because it's not about what drug makes money or what drug does this or that. It's about, can we find ways to give patients and parents hope and stress relief? And that is so, so important. That is, because every time I know it's important, because every time we get it in one form or another, it's such a relief and it's such a weight off of your back for a minute. And it gives you something to believe in. It gives you something to attach to and go, oh, Dr. Berry said that it's not a pie in the sky hope that gene therapy could cure galactosemia in the next 20 years. And I remember when he said that, what I felt. And I felt like, holy shit, if the smartest person in the world to me thinks that that's possible, that's hope. That's very real, tangible hope that I can grasp onto. And so having that be the intention behind all of your research and like your motivation behind what you're doing, I think is not only like admirable, but it's like super, super important. And it is really hard when like you wish you could give these parents these things, like you could give them all of these tools just immediately and give them this relief. But it oftentimes probably feels like a slog of doing research every single day, and you're making these tiny, tiny little steps, and you put out a paper, but then it's like, what's the next step after that? And it's like, I don't know, maybe it dies, or maybe it lives on, or maybe it goes. But just that effort is enough. Like just that effort in like doing that is so impactful and so powerful, because you're letting the community know that like they have somebody fighting on their behalf. And so as somebody in the community, I would just say thank you for like all of the work that you have put in and that you continue to put in. And I'm sure the community of diabetes and women who are pregnant and can't have carbohydrates would say that same thing for the work that you're currently putting in. That's a very long-winded way of just saying thank you for what you've done. It's so appreciated by our community.
Well, you are welcome. I was just thinking how there's not much glory in in researching rare disease, but there is that human element that is really important to me, and just working with people and with parents and families. And I think that was one of the reasons I was really excited about purple sweet potatoes, because it's something that you can get a little bit of control with and have that little bit of hope, as a parent, just feeding your kid purple sweet potato, it's not hurting anything, and it's not be helping a lot of things. So
it might be helping a lot of things. And if you need somebody else, it's like, well, are they doing it? I can tell you right now, we do it with Elliot. I make him purple sweet potato every chance I can get. And sometimes it can be hard to find. I think, my wife's on visiting family on Long Island right now with Elliot, and they're having a hard time finding purple sweet potatoes. So you got to like really do some digging to find it. And then once you find that place, you got your place for purple sweet potato. And it's where we go to Sprouts all the time and get them. So it is that little bit of hope at least helps.
Yeah. And I can just say, to relieve a little bit of your stress, if you're traveling and you can't find it, strawberries, blueberries, any of these colorful berry, cherry, have similar, if not the same, compounds in them. So it's just that purple sweet potato is a dense, cheap source, relatively speaking. Okay. So
good to know. Great to know. Synneva, thank you so much for joining us on this show. You are welcome back whenever if you ever want to come back and show us a new piece of research that you're excited about, you're always welcome. I really appreciate you talking about two topics that I've wanted to talk about for a long time, and I know specifically with POI, there's definitely people out there who are very interested in that topic. So I appreciate you taking the time and giving us your thoughts and lending us your wisdom. I appreciate it, and we'll talk to you soon.
Wonderful. Thank you so much. Talk soon.